So I asked you to do something for me before class started, and that was to watch a sliding filament theory video or two. Because that's where we're headed today. We're going to start talking about this mechanism in which we believe the muscles utilize to contract and relax. And there's a lot of steps, unfortunately, so it's a process. And we're going to go through the process like 2 times, slowly, so that you really have a good understanding of what's going on. So before we get there, let's do a quick review from last time. This was one of the most important slides from last time. We said that this is a skeletal muscle cell, or a myofiber. And we said that there were some unique characteristics associated with this cell. In addition to the things that we learned from earlier in the session, we said that it is multinucleated, it has the prefix sarco, there's glycogen and myoglobin, and gigantic mitochondria. And then we talked a little bit about the triads. We're going to talk about triads today, and we're going to get into this whole business of thick and thin filaments. So when I say thick, hopefully you're saying myosin, you're saying golf club, and when I say thin, you're thinking of the beaded necklace, and you're thinking of the relationship that they have with one another. And we said that the organization of these thick and thin filaments is represented by a sarcomere. And so each sarcomere contains one block of thick and thin filaments. And as you can see, that is what creates the stripes or the striations that we see in skeletal muscle, one of the major characteristics. So this is kind of where we ended. You should be able to talk about what a sarcomere is, and you should be able to talk about labeling or drawing it. Remember we drew that on the board. And then as far as today is concerned, we're going to get into kind of how the sarcomeres change shape. Do they stretch, do they contract, or do they just slide? And so that's where we're headed. Now before we get there, we have to talk about one component that we're going to talk more about after this next test. So this test, which is next Thursday, is considered the muscle test. After the muscle test, we're going to spend the rest of the semester talking about the nervous system. So the brain, spinal cord, nerves, things like that. So what you see up here on the right is a slice of the spinal cord. And when we think of the spinal cord, we think of a place that allows for information to travel towards the brain and away from the brain. So sometimes people call the spinal cord an information highway. It's going in both directions. So information that is traveling towards a muscle is what we see here. And if we blow up these ends of these axons, we know that they're called neuromuscular junctions. We saw a model of that in lab. That was on the lab test on Tuesday. It'll be on the lab test tonight. So a neuromuscular junction is the communication area between a nerve and a muscle fiber. But one of the things that we have to emphasize is that when we have communication in clusters, we refer to this whole cluster as a motor unit. And remember what we said back, 3 lectures ago? We said motor units are located in the belly of the muscle, the thickest region of the muscle. So think of like your biceps, the bulge of the biceps. That's where we have most of the nerve communication with that particular muscle. So motor units. Sometimes the motor units are numerous when we have things like fine motor control, like fingers and hands. Some of you might want to pursue a degree in like occupational therapy. You're going to be dealing with people who have hand injuries and trying to fine-tune their fine motor units, right? So sometimes the motor units are in charge of some very large and impressive muscles, like the gastrocnemius muscle. That's a very big muscle in our calf. So that's one of the muscles that allows us to walk and jump and do things like that. So just remember, neuromuscular junction, clusters of them are called motor units. And I think that's kind of the theme that I want you to know for the next test. Here's an actual picture of a skeletal muscle. You can tell it's a skeletal muscle because of the stripes. And you can see the nerve endings kind of not attaching, but communicating. Now in lab, we started to talk about neuromuscular junctions. And we said that that model represented a knob, and it represented the membrane of the muscle cell. And one of the things that the model had a hard time doing is representing that both of those, the nerve and the muscle membrane, never really physically touch each other. So let me skip over this slide and just show you a picture. And let me put the slide down. I think this would be a good one to take a picture of. This represents that model that we saw in lab. So let's spend some time talking about what we see here. First thing up here is we see part of an axon. So you might remember from earlier in lab, we talked about neurons having these long tails called axons. You can see that axons are surrounded and covered by a protective covering called myelin, which is a big topic for our third test. But at the base of the axon, we have this knob, and that's what was on the model, right? We said that only thing that you have to know from the lab test tonight and on Tuesday is that this represents the knob, and there's a bunch of sacs in here. Sacs containing a chemical that excites muscle. So we said acetylcholine was found in each one of these little sacs. You can see some mitochondria and some other things, but notice there's no physical contact with the sarcolemma. Remember, the prefix sarco refers to muscle. So here we have the nerve component. Here we have the muscle component. Let's freeze tag here. Notice the muscle component. We see more than one nucleus. We see very many large mitochondria. And down below here, within the sarcoplasm, we see some bundles of proteins. We call those myofilaments. And you can see, look, a Z disc and a Z disc. You can see a band and an I band, dark band and a light band. So everything that we see here is something that we've talked about. But one of the most important parts of today's lecture is the fact that on the sarcolemma, we have, what color is this thing, purple? We have these little purple rectangles, and these represent receptors. So when we have communication between a nerve cell and whatever, you have to have receptors. And the receptors have to be specific to these little specks in these sacs. So there's lots of specks in the world of the nervous system. There's lots of different neurotransmitters. Not every type of neurotransmitter will interact with these specific purple rectangles, the receptors. But luckily, muscles have, look, acetylcholine receptors. Here we go. ACH receptors. You can just write ACH on the exam, acetylcholine. So this sets us up for something really important, and that is how does the nerve or the brain send a signal to a muscle to contract? Because in some cases, the muscles are voluntary, and we can go like this, and we can stop, and we can go like stop, right? But in involuntary muscle that's involved with digestion, we still have this neuromuscular junction, but it's happening behind the scenes. Same thing with cardiac muscle. So this is an important figure. Does everybody feel comfortable identifying like some components of the skeletal muscle, the knob, the vesicles containing acetylcholine? Okay, everybody good? So this is a good review for the lab test tonight as well.
Mike?
Yes.
Quick question.
Yes, ma'am.
You called those sacs vesicles, so is that the same thing?
Yeah,
Okay.
so a vesicle and a sac are the same thing.
Okay.
So these are just containers, you can see it right here, synaptic vesicles containing acetylcholine.
Okay.
Could you
Yeah.
go back one more? I'm sorry.
Sorry, go ahead.
Could you go back one more slide? When, oh, sorry, yeah, the other back, one more with the picture.
Oh, we haven't gotten there yet.
Oh, the other back, the real back,
This one?
this still back to the other picture. So when that
This one?
clicks right
Okay.
there, is that
Okay.
one axon terminal button down there?
Oh, that's a good
Or is
question.
that a bundle? I'm trying to like think size.
So this is an axon, this is an axon, this is a third axon.
Okay, so
Yeah.
that is just one end touching one fiber.
Right.
Okay, cool, thank you.
Yeah.
That would be the knob, right, that darkened area.
No, this is probably the nucleus.
Oh.
Yeah.
Oh, the specs?
This circle right here.
Yeah,
That's
that's
probably the
the,
nucleus.
oh, the whole circle's a nucleus?
See, no, these
I
little
thought those were
specks,
the-
these little specks are the nucleus. Yeah, so this is probably the neuromuscular junction.
Okay,
Yeah,
okay,
yeah.
cool.
Okay, good. All right,
Thanks,
so,
Mike?
let's, yes,
Sorry,
Joe.
is the mitochondria that's in the axon terminal,
These?
are they producing ATP to power what they're doing and then?
Yeah, so this, believe it or not, this knob is still part of the neuron, which is way back over here.
Right, that's why I wondered.
So this is still part of the cytoplasm. So it has to, everything in the knob is also found in the cell body. So it's trying to keep the cell alive.
Okay,
Yep.
And then what did you say was short, sarco was short for
Muscle.
muscle?
Anytime you see sarco, think of muscle. So sarcolemma, we said sarcoplasm. Anytime you see sarco, think of muscle.
Fine.
Yes.
So,
Nicole.
so, like, I'm sorry, it's for you. Oh, okay. I'm sorry. So somebody with like Parkinson's, is it a misfiring of these, or is it?
That's a great question. So Parkinson's is a situation where we aren't able to release a neurotransmitter that kind of quiets muscle contraction. So you can excite it, but you can't just quiet it. And that one you may have heard of is called dopamine. So dopamine is something that is not released in the quantities that it is in you and I. So we can move muscles and then we can tame them and slow them down, but not in people with Parkinson's. We'll talk about that a lot. Okay, so here's kind of what we just looked at, and we're trying to figure out. So here is the axon, here is the synaptic knob with the neuromuscular junction, and it's trying to make a connection. And remember, the cell body with the nucleus is really far away. All right, so now let's get into some craziness, part one, which means drawing. So what I'm gonna draw right now is probably gonna be one of the most important things for the rest of the semester. It's also gonna be one of the most important things for next semester. So this is really, really important. So the first thing that I'm gonna draw is the muscle membrane, the sarcolemma. And remember, this is the membrane of the muscle cell. And outside, this is outside, there's fluid that surrounds the cell. And on the inside, there's fluid that surrounds the cell. So ECF stands for extracellular fluid. That means fluid on the outside. And ICF really refers to the cytoplasm, in this case, the sarcoplasm. Okay, so now we know that there's two fluids on either side of the membrane. Now let's do this. We're gonna say on the outside, there are a lot of ions in the ECF. There's a few anions, but the majority is positive cations. And that cation that's most prevalent is sodium. So right now, you know that on the outside of the sarcolemma, we have a lot of cations, which are positively charged. And on the inside, we have some positive cations, but the majority is negative. So the take-home message so far is that this pore membrane that we're looking at is kind of stuck in between two drastic different charges. So on the outside, it's very positive, and on the inside, it's very negative.
Does it have a lot of potassium?
On the inside, there's some potassium, yep, there's some potassium, there's some chloride, there's a lot of different things in here. There's some phosphates. So, now that we know that the membrane's stuck in the middle, one of the things that we have to talk about are some terms. One is a term called resting membrane potential. And that's what we drew right here. It says that at rest, when a muscle is not contracting or relaxing, just kind of sitting there, it's stuck in between two different charges. And the fact that it's in between two extremes means that it is electrically, ready, here we go, electrically unstable. And that's a good thing, because when something's electrically unstable, it can react quickly. So it has a lot of, look, potential. All right. Now, we're going to talk about a series of events that gets muscles to contract. But one of the things that happens several times is that we have a brief shift in this resting membrane potential. For example, here we go, watch. There will be sometimes gates in this membrane that will open. You might remember us talking about the plasma membrane, the phospholipid bilayer. You drew that on test one, the circles with the two tails. We said that there's a lot of proteins in the phospholipid bilayer. Some of them act as channels. So in our story today, we're going to be talking about situations, look, where we have lots and lots of positivity rushing in. Yeah, it could be an example of a gated channel. That's definitely one of the things we're going to talk about today. Yeah, so a gated channel normally stays closed, but occasionally something triggers it to open, and that's definitely going to be the case today. So here we have a situation where a channel opens that allows the positive cations to rush in. This process right here has a big term. And underline this and circle this, because you're going to hear this probably 20 times before we leave on August 8th, and you'll hear it probably 10 times next semester. That process is called depolarization. So you could simply write down in your notes that depolarization occurs when the inside of the cell becomes positively charged. When the inside of the cell becomes positively charged. We also talked, I think, on lecture 2 about homeostasis. And we know that cells have a set point. And being positive is not a part of being its set point. So almost immediately, another channel will open and kick out all of these positive cations, returning it to its normal resting membrane potential. We call this process of kicking it out repolarization. Now, if you can leave today and say, I know what depolarization and repolarization is, I'm going to call tonight a success, because this is like one of the most important things in physiology. We'll talk about this in the way the heart beats and everything. Depolarization, repolarization. But what's the big deal? Why does this happen? What's the purpose? When we have a depolarization followed by a repolarization, it generates an electrical spark. And that electrical spark is called an action potential. So an action potential is created when there's a depolarization followed by a repolarization. I want you to just stop and say that like 3 times in your head so you remember that. Otherwise the story of what we're going to be talking about tonight is going to be crazy and going to be really, really hard. An action potential is created when there's a depolarization followed by a repolarization. Sometimes people will refer to action potentials as nerve signals, brain signals, or electrical energy. But now you know how it's triggered. It's this shift in charge. Okay, so that is really important to understand these two. Does everybody get this?
You said that maintains homeostasis?
Well, when we make something positive that normally isn't positive, it's going to try to maintain homeostasis and kick it out as soon as possible. So homeostasis causes these two events to happen really quick. Okay, so good. Let's get back to the slide. When we think about now what we learned in terms of generating an electrical signal, we can start this and begin to start thinking about what happens inside the sarcolemma. Okay, so the plasma membrane is polarized or charged. Yep, we know it. We drew it up here on the board. It says the outside has a lot of sodium. On the inside, there's a lot of anions with a little bit of potassium. And I'm not going to have you memorize this number, but if you would measure the amount of negativity on the inside, it would be this number. And I'm not going to ask you to memorize that number. So when we open and close the gates in the sarcolemma, we can create sparks. Sparks are what's necessary to create a muscle contraction. So here's this. This kind of shows maybe what I kind of drew. So here's the plasma membrane, the sarcolemma. Here's a bunch of sodium on the outside. And you can see that we have channels. Sometimes the channels will allow positivity to enter or negative to leave. It depends upon the channel. And Michael was good in that he was able to pick up on the types of channels being called gated channels. All right, so let's go through the process now of talking about the steps for a muscle to contract. And I think what we'll do is I want to tell you a story without you taking notes. And just kind of look and listen. You might catch only 20% of the story. But then I'm going to repeat it and I'll go slow and you can jot some things down. So this first attempt is just like, okay, let's hear what's about to happen. You might soak some things in. So the story begins with the neuromuscular junction. This was exactly the model we saw in lab. And this arrow represents a nerve signal, or you could say an action potential, right? An electrical stimulus. So what happens here is that the electrical stimulus comes down, watch this, and it interrupts the membrane of the knob in that it opens up special gated channels. Now, remember, a gated channel stays closed unless something triggers it to open. The channels that are in the knob are called voltage-gated channels, which means they stay closed unless they get zapped. And that's what this arrow indicates, an electrical zap. So when these channels feel that electrical energy, they open up, and the channels only allow calcium to come in. What? Calcium, what we thought was good for bones. But now we know that without calcium, you can't have a nerve communicating with a muscle. So calcium rushes in, and the purpose of the calcium is to trigger the release of acetylcholine. So it tells all these sacs, all of these vesicles, to move to the edge and to dump and release all of their chemicals in this gap called the cleft. You guys remember what that process is called? Not when you're eating something, but when you're spitting something out. What is it? Exocytosis. Right. So when we are eliminating something, we're saying that those vesicles, look, move to the edge, look, move to the edge, and all of the acetylcholine is released inside the cleft. And luckily, right, on the other side of this space, there is the sarcolemma that just so happens to have receptors for acetylcholine. It's perfect. Okay, so that's page 1 of our story. Is everybody
Why is
okay?
that calcium with the synaptic knob? How's that happen?
Well, calcium is out here in the extracellular
Just in the,
fluid. Yeah,
okay.
so you have calcium, all kinds of stuff in the ECM. Okay, so let's take a look at this thing right here. This represents one of those little purple rectangles. And this stays closed 99% of the time. But if it binds to acetylcholine, then it's going to open. So this is a channel that is also gated, but it's called a ligand gated channel, not a voltage. It says, I will only open when I attach to some type of chemical. That's what the word ligand means. So when the ligand or the acetylcholine attaches, then look what happens. Well, this is the picture from your book. You can see that you have these vesicles kind of moving and they're binding. And this is all the membrane of the muscle. So once they bind, look what happens. They open up the channel. And look what happens. Sodium rushes in, potassium leaves almost instantly, and it creates a spark. Whoa. But I'm looking at 1. Remember what we said is that in this membrane, there's thousands of these receptors. And when this occurs, when we create our first initial spark on the membrane, it's enough to make the whole membrane electrically excitable. In fact, we call the first action potential something called an end plate potential. It's the most impressive one because it's happening almost at the same time. So what's the big deal about the end plate potential? Well, when it creates that spark, there's nearby, look at this, in the sarcolemma, not ligand-gated channels, but there's nearby voltage-gated channels. That spark is enough to zap these guys, and look what happens. Sodium comes in, potassium rushes out, guess what? That creates a spark. And that spark excites the next voltage-gated channel next to it. It's like a series of dominoes in a line. The initial domino was this end plate potential. And then there's hundreds of voltage-gated channels on the sarcolemma that get stimulated, ready, by an electrical volt or an action potential that was created by the one next to it. So it's just like it's like dominoes. So take a look over here. You can see that here's our knob, and as we create this electrical signal, this arrow business representing the electricity, it continues down the sarcolemma until, here we go, it meets one of the components of the triad. Remember the 3 components? We said two cisterns, right, that store calcium, and then 1 T-tubule that we said was an electrical pipe. Well, look, that electrical pipe takes that electricity and it forces it down, and it causes all of the calcium in the cisterns to leave because the cisterns have voltage-gated channels. Oh my gosh, it's all about electrical current. So the cisterns release their calcium because they've just been zapped. And guess what happens to the calcium? Oh, this isn't the best part. The calcium binds to the little triangles on the spaghetti. Remember what those were called? Troponin. And as soon as calcium binds to troponin, look what happens. The spaghetti molecules, the tropomyosin, swing over, and look, there are the active sites. And myosin is getting happy. almost excited, like, I can go in there. It's the first time I've seen it, right? So without calcium being released, these active sites would never be exposed. So all we have to do now is put in the golf club head into each active site, and they all get filled. And then something called the power stroke, which I'll talk about in a minute, occurs where the golf club inserts, and then they all go like this, like that. Which means they grab a hold of these beaded necklaces and make them slide. And when it slides, thanks to the power stroke, every myofilament moves towards the center. And remember what I did with my thumbs in the last class, is that when they slide toward the center, the distance between my thumbs, the Z-disc, gets smaller. So how does it slide? It happens because the myosin golf clubs can find the active sites, and they all have a power stroke at the same time. So the power stroke allows for that part of the contraction, and that's what makes muscles in general smaller, is the sliding of this. Okay, so that's it. That's the whole process of contraction. Let's go back and let's go slow. Ask me questions, take notes. And this is again why I told you to watch at least 3 videos on YouTube that explains this, because sometimes seeing the whole process actually takes place.
Mike,
Yes,
is there any
yes.
way you put the screen down so we can get some?
Yeah, I'm gonna do the screen down now, because I thought I was gonna draw on something.
Okay, thanks.
Hey, I'm gonna go talk to Sean real quick.
When do you talk about closely recapping all stuff?
Okay, so let's get this party started. It starts, ready? It starts with the neuromuscular junction. Your brain is telling your skeletal muscle to contract. So the brain is sending an action potential. That's what this arrow indicates. As soon as the electrical impulse gets to the knob feels that electrical voltage and opens up, remember, voltage-gated channels. And calcium floods in. What is the significance of calcium? We said that calcium triggers exocytosis, where we have the release of acetylcholine into this gap, the space, which we call the cleft. So, once we have acetylcholine in the cleft, it then looks to bind to acetylcholine receptors, which are in the plasma membrane of the muscle, or the sarcolemma. Remember, the sarco means muscle.
Are the
Yes.
ACH receptors only where the neuromuscular junction is? Or are they
Yes.
also- Okay,
Yes.
they're not other
They're not,
other
they're
places?
not, yeah, because like over here, you
Yeah.
don't see them.
Okay.
So they're only in the cleft. So once they bind, we said that it opens up this channel. And this is not a voltage-gated channel, but this is a ligand, L-I-G-A-N-D, a ligand-gated channel. And remember what a ligand is. It's the official name for some type of chemical, any chemical that binds to a receptor. So in this case, the ligand is acetylcholine. When the ligand attaches to the receptor, since it's a gate, says, okay, I'll open. And we said there's lots of these receptors, and they allow sodium in, followed by potassium out. which I hope you remember is depolarization followed by repolarization. That event creates an action potential, an electrical spark. And the first one we said was impressive, so impressive that it has its own name. It's called the end plate potential. So on the test, you might see this, EPP. What is an EPP? It's the first electrical spark on the membrane of the muscle. That EPP is so impressive that it triggers, look, nearby gated channels. Notice these aren't ligands. These are voltage gated channels. That end plate potential causes these to open. And we have, look, sodium rushing in, potassium leaving. And that creates a spark, which then causes the voltage gated channels right here, where my pointer, to open and close. That creates a spark, which then opens these. So it's just like we're moving the electrical signal down the sarcolemma until it reaches a T-tubule. Remember that? So once it gets to the T-tubule, it flies down. And that electrical current causes the release of calcium, which is being stored in the cisterns. So right here, everybody look, this is a triad. One T-tubule, two cisterns. The cisterns are full of calcium. All right. So the calcium is released in the sarcolemma. It has no idea where it's going. But if it finds a receptor, we know what the receptor looks like. It looks like a little triangle. We called it troponin. We drew this. Remember, we drew this. If calcium binds to troponin, it causes this big loop of spaghetti called tropomyosin to swing over and to expose on the beads. You guys remember what these beads are called? G-actin, remember that? It exposes the beads, the active sites. And these are the perfect shape for the heads of the golf club. So now we can see how myosin and actin potentially can interact with one another. Does that make sense so far? Does everybody understand how the active sites become exposed? Only by calcium being present. So if you have a situation where you don't have if you have really low levels of blood calcium, you may not get a muscle response. What? You could die because you have low calcium in your blood, because it's going to affect your muscles. So let's talk about the interaction. We said that the golf club fits into the active sites, and it does this kind of cocking and releasing that I'm going to call the power stroke. For this class, I'm just going to say know what the power stroke is. You do not have to give me any details. Everybody hear that? You don't have to give me any details. Just tell me that once the myosin head interacts with the active site, a power stroke occurs. That's good enough for me. But it involves an energy molecule called ATP. So, one of the big essay questions on Thursday's test, is asking you, how is ATP used? So let me give you half of the answer right now. ATP is used in contraction to generate the power stroke, which means inserting and grabbing and sliding. ATP is used for the power stroke. That's all you have to say on the test. You'll get it right. ATP is used for the power stroke. So here the power stroke, you can see ATP is on binding to the myosin golf club. It utilizes ATP to push and slide the filaments past. So you can see the whole process of the golf club interacting with the beaded necklace, and it kind of pulls. You can see the arrow causing a shift in the filaments. But what happens then when the muscle is finished contracting? This is the next part of the story. Notice that there is no, look up here, there's no arrow of electricity. The brain says, I'm done. So if there's no arrow, look, then there's no more calcium. If there's no calcium, it means, hey, let's kind of close up shop. And so we try to suck some of the acetylcholine back in. You might remember the process called endocytosis. So with calcium, it's exiting, exocytosis. Without calcium, it's endocytosis. So you can see the arrows saying, come back in. But there's still some residual acetylcholine lingering in the cleft. And we utilize a very special enzyme. Look at this enzyme. And look at the shape of it. This enzyme is called ACHE. This is called acetylcholine esterase. So that's too long to say, so just put ACHE. And ACHE gets rid of any acetylcholine that's still in the cleft because we want to stop the contraction. So look what happens to the ligand-gated channels. They don't have their ligand anymore, so they're closed. No more end plate potential, no more spark. So without any spark, do you see any, look, arrows going down into the cistern, into the T-tubule? No, nothing. So without electricity, we utilize ATP a second time. And this is the second part of your essay question. Remember the essay question? How is ATP used in the sliding filament theory? For contraction, it's used for the power stroke. But during relaxation, here we go, ATP is used to actively pump calcium back into the tanks, into the cisterns. So calcium wouldn't want to do that naturally, but there's a pumping mechanism that requires ATP. If we don't have ATP present after we release calcium, guess what happens to the muscles? They might stay in a locked position. Guess what that's called? Rigor mortis. No, 100%. That's what rigor mortis is. So when you see the animals on the road, you know, the raccoons and the deer or whatever, and they're like this, you know, they're like stuck, you know? It's because they had calcium out here, but there was no ATP to pump it out, so their muscles are gonna stay in the locked mode, contracted mode. Yeah, Mary.
What about someone that has like a contracture,
It's a great, you mean like a muscle twitch?
like it's like stuck, like,
Yeah.
no.
That's called tetanus. And
That's
so tetanus, so lockjaw or tetanus is a situation where you have a maximally sustained contraction. And that can be because of a bacteria, or it could be because of a problem with this process here as well.
Are people like not using their muscles, like with elderly people, how they just fall up?
I don't know about that.
Is that the same
Sometimes it's also kind of like a Charlie horse, you know, you know, sometimes you wake up in the middle of the night with a Charlie horse in your calf or your toe. That usually means that they're low in electrolytes, but then we're.
I was gonna say, is that the same with like a seizure? If someone's having a seizure and like locks up and they,
Yeah, those
you're
are all
not
great
supposed to like
questions.
move.
Yeah, those are all great questions. So if someone's having like a grand mal seizure and they're down and they're like kind of locked.
Like so tight, right?
Yeah, it's probably similar to tetanus.
Okay.
Okay, so let's talk about the relaxation process. Let's rewind. No electrical signal, no calcium. Endocytosis says let's suck it back in. Acetylcholine esterase says I'll chew up any residual, any remaining. No electrical current going down in between the cisterns, so that triggers active transport. So that requires ATP. So your answer again is ATP is used for the power stroke, and it's used to actively transport calcium back into the cistern. So look what happens. Without calcium present, the loops of spaghetti, the tropomyosin, starts swinging back and covering up the active sites. And myosin is left without any opportunity to interact. So that's it. That's the whole story. Yes?
With endocytosis, is it
sucking ACH back in or the calcium?
No, just the ACH.
Just ACH.
Yeah. And we don't want calcium because that would trigger exocytosis. Yeah, Dave?
Does the, wow, not really clear. The Myzin,
Uh-huh.
when it connects with, when it connects, does it actually disconnect or is the Myzin used up? And that's-
Yeah, so the myosin never gets used up, it just literally gets disconnected. Yeah. So we talked about rigor mortis now, we know kind of what to experience and how to explain that. Again, it's the fact that we don't have ATP present to pump all that calcium away. Sometimes, and maybe you've heard of this, and some of you were kind of getting towards that with your, kind of clumped hands. This is a disease called dystonia. I want to talk a little bit about dystonia. Dystonia is a disease in which it causes the muscles to contract maximally, but completely out of the will of that person. And it only affects skeletal muscle. It affects it so bad that a person can barely walk, and they can't really talk because of skeletal muscles that are very close to their larynx and trachea. So when you see someone like they're going like this, I mean they can barely walk. Poor things. And one of the most interesting things about dystonia is that when they go to bed, their body completely relaxes. When they when they fall asleep, their body looks like yours and mine. So what it really is that it's a brain issue, dystonia. It's sending too many signals to contract. That action potential is overwhelming the muscles. So sometimes people with dystonia get something called deep brain stimulation. You may have heard that also being used to treat people with Parkinson's. They're trying to slow down the muscle contractions. It's the same thing with Parkinson's, right? Uncontrollable muscle contractions. Let's try to slow them down. It's a brain problem. And so when we talk about the word neuromuscular junction, it kind of comes into play here. So dystonia, maximal sustained contractions. Okay, last 4 or 5 slides of the day are going to deal with something that's going to lead us into Tuesday's talk. And it has to do with the length and the tension of a sarcomere. The length and the tension of a sarcomere. Now I'll come back, but I want to show you a picture first. Take a look at this. This graph down here tells you that this is in the dream world and the optimum contraction of a muscle. And it says in order to get the optimum in this color right here, you can see that, and of course you would never have to memorize numbers, but the sarcomere, the distance between Z discs looks like this. If you are overly contracted, look, it looks like this, and the response is not nearly as impressive. Or if it's overly stretched, it gives you this response, which is not at its peak result as well. Let's make sense of this. So we're saying that when you're doing some type of physical activity, you may want to stretch before you do it to get the sarcomeres at its optimal length. If you get out of the car, remember what I said, I've run like 12 marathons, and every marathon I just got out of the car and got on the starting, and I go. I don't even stretch, I don't even bend over, I don't do jack. And it's always worked out for me, never been hurt. Well, I'll get hurt tomorrow. But when I get out of the car, my muscles are looking like this, And my first 10 minutes of that run is not very impressive. So maybe I should stretch. And when I coached my kids in sports, we always stretched. But I just hate stretching, Oh, I can't stand it. I'm not flexible, maybe that's why. But let's say you do something strenuous for a long period of time, some yard work, or whatever, and you say, man, I feel exhausted. Oh, but I only have like 20 more bags of mulch to lift. Well, guess what? Those 20 bags are going to be the biggest struggle because you're overly stretched. So where should we be? Somewhere in the middle. Let's go back, and now let's take a look at this slide, and hopefully it makes sense. It says that overly contracted give us a weak response. Too stretched give us a weak response. So the optimum resting length is one that provides us with the best result because it's somewhere in between. So that in itself should convince me to practice what I preach. You know, like, I should probably stretch. I should do some light jogging or something before I run. Maybe I'll do it. Yeah, Bria.
Is that the same as when you lift and your muscle fails? Is that being overloaded?
That's more like fatigue.
Okay.
And that's something we get to talk about in detail on Tuesday. Yeah, Tuesday's talk is all about exercise, so that should be kind of fun. All right, couple terms and then we're gonna call it a day. I would like for you to redraw, no. Go ahead and take a picture of this. This is a big one. Let me give you another big essay question on the test. So I've given you one, he'll give you the second one. Is that if I give you this graph of one muscle contraction, we call it a twitch, you should be able to label a muscle twitch. You can say that it's getting ready to contract, or you could say resting membrane potential. Then you can say, oh, the muscles are getting shorter, they're contracting, power stroke, and then they're relaxing. So what I would like for you to do, here's the question, is to summarize that sliding filament theory using this graph. And write some of these terms down. I want you to make sure that you include words like calcium and depolarization and repolarization. Of course, you should be including ATP in that conversation. So when I say latent, you would probably say resting membrane potential and explain what that is. When I say contraction, do you have to explain every step? Summarize it. Talk about what happens in order to get the power stroke. And then relaxation, summarize it in like 3 sentences. So the whole thing should be like one healthy paragraph. I'm gonna give you 1/3 of a page of paper to answer it. So if you practice writing this and it's like a whole page, that's too much. I want 1/3 of a piece of paper full of information describing what we've been talking about for the last hour, okay? All right, now when we think of muscle twitches, that's not a very common type of muscle movement. And so in most cases, muscles work together. So they work with several other muscles. In some cases, some people try to explain muscle fibers as being muscles that, you know, contract maximally if they're stimulated. But we know that it's not true because every muscle has different fascicles. And so we can get different responses. One of the things I always think about is my backpack is pretty heavy, but I bet some of yours is heavier than mine. If I had 2 backpacks in my hand, I'd say lift them. Or if one backpack was completely empty, I'd use the same muscles to do a curl, but if one is really heavy, I'd have to recruit more muscle fibers, more muscle cells. So don't tell me that a muscle contracts maximally, otherwise when you pick up a gallon of milk, you'd be slamming your head, you know, whack, you know. So take a look at this, and there's a word here that will be important for our last exam with the nervous system, and that is threshold. So what the dotted line indicates is just the amount of electrical energy or action potentials that's necessary to get a muscle contraction. So notice, here you can see the stimuli to the nerve, that in this first 2 stimuli, it's not enough to release calcium. It's not enough to get a response. But once we start releasing more, we get a response. We see a little twitch. That's what we just drew on that last graph. And then you can see that it continues. But what's interesting is that every muscle has a maximum. A maximum. And beyond that, you can send as many signals as possible, but you're still gonna get the same response. So, you know, whatever that is, whether it's like this, you know, or this, you know, it's a maximum. You can't do any more than that. So there's always a limit. Notice this represents back to back to back twitches. It's kind of like walking. But this kind of represents running, where there's very little time to relax. And so once we have this kind of response, then we start thinking about how do we utilize different types of energy. Remember we said the sarcoplasm contains glycogen and myoglobin. So that's the process of creating a movement. So we have like 3 slides left, and I'm sensing that we have already covered enough. So I'm going to start with this. No, I'll go back. I'll start with this slide on Tuesday, and we'll finish the last 3 or 4 slides. Then we'll do our last talk on Tuesday. So I want to give you some time. So you're going to have 25 minutes to kind of get into lab mode. I will open up the lab door at 6:30.